Okay, I think it works now. Hi, hello everyone. Microbehunter here. Okay, I always have to wait a few seconds at the beginning of the live stream because there's a short delay. And I think everything seems to be working now, at least I hope so. Okay, yeah, hello and welcome to another Saturday microscopy live stream. Sound is a little quiet. That's interesting. Video and sound. The thing is important. The sound is important because as you might see over here, I got myself a new microphone. Yeah, it could. I mean, the audio indicator actually says that it should be loud enough. I mean, I could make it louder, but most of you are saying that it's fine, so I'm quite happy. Otherwise, please, yeah, quickly write in the chat. If it's too loud, for example, then I can tune it down a little bit. But again, as I just said, this is a microphone. It's the first time that I'm trying it out. Yeah, and yeah, I'll see how this works. I don't know if I try to move it away a little bit further. Yeah, if you feel it's too quiet, then please tell me. Okay, great. Okay, first of all, yeah, here we are again. I'm really happy that you are joining me again this week. You probably have already seen from the thumbnail of this video, of this live stream, I just called it microscopy live stream, and I did not talk about any. I did not put any specific topic on the thumbnail and also on the yeah, on the title. And the reason is because I would like to try a slightly new format. Usually in the past I've had one main topic, maybe a few smaller ones as well. But I want to try the following. I just want to do several shorter topics that are microscopy related. Okay, sometimes completely disconnected from each other. But I hope that this way, to have several shorter ones, maybe I'm able to cover your interests better. Okay, so that's kind of something I would like to try out. What I also would like to say is that some of you already started to post questions even before the start of the live stream. So, for example, there were two questions here that wanted to know about my camera setup. And I said, that's cool that if you want to know things like this, then I can prepare in advance. So if you have any specific questions that you would like me to talk about in the live stream, please post them. And if you post them in advance, then maybe I can do a little bit of preparation as well. Okay, so no guarantee that I'm able to answer the questions to your satisfaction, but in any case, that's a little bit the idea. Yeah, I'm going to, as always, because it usually takes a couple of minutes for people to join in, I'm usually going through the chat here a little bit and to say hello around the world. So there's a hello from Vietnam, from New Zealand, okay, London, UK, Belgium, Norway, okay, Rhode Island, USA. Sound seems to be okay. Arizona, USA, Birmingham, UK. Okay, sound is fine. Nicaragua, Canada. Oh, I love it. I love this international community here. Hello from the UK. So a question about the AmScope. I don't know this one. So what I will be doing, Montreal, Netherlands, and hello from India. So what I'll be doing is the following. I will simply go through the questions later on a little bit, and then I will move on to the next section. And this one over here, believe it or not, is chocolate. And I'm going to put it under the microscope as well. There will be a separate video about this here, but I'm going to unpack it. I'm going to put it under the microscope because, yeah, it's going to be nice. I hope I don't forget about this. And what I'm going to do is because I promised, several of you wanted to know a little bit about my camera setup. And here, this is already the first question. This is which camera setup? The one on my microscope, the one over here on the desk. Maybe you're not so interested. The one over here, yeah. It's a webcam. So I think you're really more interested in my camera setup for the microscope. Okay. So what I'm going to do is, okay, I'm going to now use this here and I'm going to show you my camera setup that I use for my microscope, and I'm going to explain it a little bit. Okay. So let me quickly see. So that is, yeah, I have to press this here and then let me see if this works. Okay, just a second. That's the first interesting. Okay. Now it's blanked out. I do have to get the microscope to work, and this is a little bit of strange. Now it works. Okay. Yeah, so that's basically now the camera view that you have, okay? The microscope view that you have, but that's not what you want to know. You want to actually know how the camera is connected to my microscope, and for this reason, I'm going to switch over and I'm going to show you by moving this desk cam that I have over here. I'm going to flip it over, okay? And I'm going to show it to you, and this here is the camera that I've connected. It's an Olympus D90 camera. It's a DSLR camera, okay, and it's connected over this adapter tube here, and then it's connected to the microscope. So these are the eyepieces here, right? And on the bottom, I'm also going to show you this here. This is the other camera, which basically shows you the view of the stage here when you look through the microscope. So this camera over here, okay, captures. I'm just going to show you. Captures the one that you see up here, okay. So you see if I bump it, then you see it shakes. Okay, so and yeah, so that's kind of that's kind of the way it's connected. I would like to talk a little bit about this because the setup, as you can see, is quite wide here, and this basically means that, but still there are no optics in here, no lenses, no optics, because the objective down here, those objectives down here. Directly produce an image into the camera sensor up here. Yeah? And because of that, this has the really cool advantage that there are no optics in between here which could actually degrade the image quality. And so it's probably one of the more theoretically best image qualities that you can get. And why did I use this camera? And the reason is because the D90 camera allows me to connect it over HDMI. That's the HDMI cable which goes into my computer so that you can see the view. Yeah, and this cable over here, that's a power supply. Yeah? So that I don't have to use a battery. I'm using a power supply so that I don't run out of battery. Yeah? And that is basically my setup here. And why did I use the D90? And the reason why I chose the D90 is because it also allows me to make 4K video. And a 4K video is actually something that you normally don't need. Let me quickly activate this again here. Yeah, here we go. Okay, so 4K video is usually something that you don't need, and I don't have 4K here now. Right now I just use 1080p, which is also fine enough. Yeah, but still, if I wanted to, I can take also 4K video. That's kind of the setup. You're probably gonna say, wow, that's kind of expensive because the D90 camera is so expensive and so on. So there is, of course, a cheaper version over here. And that is the following, and that's if you have a microscope. Look at my face a little bit. Sometimes there are these dots in my face. That's because of the filter that I'm using. So if what you can also do is you can get yourself those adapters here. Okay, and you know what I'm going to try to do is I'm going to try to maybe change the brightness a little bit here. Okay, and what I'm going to, what you can do is you can use those adapters here, and they can be put directly also into the photo tube, and then they can be connected also. This is so-called a T2 adapter ring. They exist for Olympus and for Nikon. And then what you do is I made already separate videos of this, yeah, to explain this in detail. But then this kind of goes in here like this. Where is it? I have to find the dot. I think here it is. Okay, goes in here like this. Yeah, and then the whole thing goes on top of the microscope. Yeah? You can theoretically also put it into the microscope instead of one of the eyepieces, but the problem is it's so heavy that you have to be careful that the microscope doesn't tip over, or you have to give it some additional support. Okay, that is only one version, one way. There are different things as well. For example, you can use HDMI cameras and so on. So there are different ways how you can connect a camera. But because you were just asking about my setup, and I just wanted to show that to you. Okay, yeah. So this is a little bit. If it's an Olympus, why does it say Canon? Well, the thing is the following. That's one of the questions. I've got an Olympus microscope, but a Canon camera. Okay, so that's the thing. Huh? So the microscope is from the company Olympus, and the camera is from the company Canon. Yeah? It doesn't really matter because those adapters have to fit. That's all that's important. So just just wanted to share this with you. I mean, I've also made in case you don't know that yet, I do also have another microscopy channel. So if you're more interested in cameras and more about the microscopy hardware, please do check out also my other channel. It's called Microbehunter Microscopy. It's a very similar name. Links should be also available in the description. And if you check out that channel, then I'm talking mostly about microscope reviews, microscope hardware, cameras, and again cameras, and so on. You get the idea, right? But because not everyone's interested in cameras so much, some people are more interested in the observation part. This is why I separated those channels here. Okay, there are a couple of, I'll quickly, before I move on to the next topic, I do quickly want to answer some of the camera related questions here. So there's a question about the Amscope T490 here by Jesse. Sorry, I don't know this microscope. Okay. Okay, yeah. So that's simply something I just want to say. I don't know, you have to try it out, okay? Camera is overexposing. Yeah, that's probably because of the dots. That's actually not necessarily only the overexposing part, but because I've got a green screen in the background. And if it's not properly adjusted, because the blue, dark blue background that you see actually is not the real background, it's actually a green screen that I have. And if this is not properly adjusted because of the brightness, then sometimes you see those dots here. Yeah. Yeah, so the hello goes on from Montreal, Netherlands, Connecticut, USA, Australia. So it's Jesse saying, Oliver is speaking of cameras. Have you seen the Switch Lens project on Kickstarter that gives a high quality Micro Four Thirds sensor for mobile phones and PC for a very good price? I don't know it, but thank you for mentioning it. Okay. Yeah, so this is, yeah. Is there any community or group on Instagram, Facebook, or any platform that's working on protist diversity? Oh, I don't know about that. I mean, there are honestly lots of folks are doing research on protists. Very often, this is also on university projects. But what I can recommend to have a look into is there is this app called iNaturalist. Like the letter I, Naturalist, and this is an app, and people can take pictures with that app, and then it uses some AI algorithm to identify, and they've got quite a few microorganisms, including protists, in there as well. So check out the app or the website iNaturalist. Okay, yeah. So yeah. So that's again the Olympus Canyon kind of thing. Yeah. Okay. Does the microscope camera use a C-mount? No. Good question. I just want to explain this a little bit, and I'm going to show you know what, I'm going to show you my other camera setup, the one that I've got on my stereo microscope. Okay, so I have to lift this up carefully. Okay, and this here is yeah, that's my stereo microscope. You see my messy table. Yeah, this is the stereo microscope, and what I have here is an industrial camera, HDMI power supply, and this here is a C-mount. Okay, I've been covering my face here a little bit. This here is a C-mount, and this C-mount has to be connected to an adapter. Okay, and I got this adapter. You can buy this separately, or I got it from another microscope camera. And then on the bottom part here, it's basically just plugs in either into the trinocular head or into instead of an eyepiece. Okay, just to show you, the C-mount is significantly smaller. Yeah? Just to clarify this, because this is also often a question that arises. Okay, so. So look, I have to be careful that I don't miss any questions here. Lots of microscope camera questions. Okay. Yeah. Okay. Yes. Thank you for the. Okay. I have to check my post because there is a comment that's for my address, and maybe I overlooked it. Okay. What's a C-mount? A C-mount is a standard which is used for industrial cameras. Actually has a longer history than that. Yeah, but it is a standard for small size connections. Okay, you know what I'm gonna do is I'm gonna move on. Okay, if it's okay for you, yeah. Um, and I hope I didn't forget anything because it just so many comments, which is nice. Yeah, I hope I didn't forget anything. Is there an adapter that I can put on my microscope so that I can take pictures with my phone? Yes, there are cheap mobile phone adapters. And basically, these adapters will simply hold the mobile phone in front of your eyepiece. So it's basically you're just taking pictures through your eyepieces, and sometimes the quality is surprisingly good. Okay, so that is indeed I would say the lowest cost option. Yeah. Okay. Which model of an Olympus microscope? My first Olympus microscope of almost 30 years ago was a CH40, a CH40, and right now I've got a BX53. BX53. You can Google it. Yeah. So I'm gonna move on. Okay, because I do also want to do a little bit of practical stuff as well, because many people are also interested in doing a little bit of practical microscopy. And I would like to show you this here again. We've a couple of months ago we already looked at it, and I would like to explain this to you. This is a seawater, saltwater sample with a lot of sand in the bottom, and I collected this. 2 years ago from a beach in northern Germany. For those of you who speak German, Wattenmeer. I was in northern Germany and for holidays, and I collected this sample here. And there were a lot of diatoms in here and a lot of movement. And it's been standing here now with a little lid covered here, so that there is not enough evaporation for 2 years. I do not expect any living things anymore. A few months ago I did check it, and there was not so much movement. But I would like to check it again, okay, and see how what we're able to see here. Okay, because yeah, I do think about cleaning up all of the things that are now standing around here, and in order to clean it up, before I clean it up, I want to really check that it's nothing interesting in here anymore. Now you see again, also here on top, on this corner over here, on the very top, there is a little bit of you see, of break-up, yeah. So these squares that you see here. So what I'm going to try to do, if you quickly wait a second, I have to make some adjustments here. This makes it worse. Now stuff starts to appear in my face. Guess I have to find some intermediate. Yeah, I don't know. Let's see how this works. No, it's not good. If I remove the thing in the corner, then something appears in my face. Otherwise, I'm just going to leave it like this. I'm going to let me try to adjust the brightness. Maybe the brightness makes a difference. Oh, what's going on here? Maybe this worked. I turned up the light a little bit. Yeah. Okay. Okay, I'll just just ignore those disturbances, please. I don't know why. Yeah, I hope it doesn't. Yeah, did microbes get into my camera? I think so. But I'm a little bit upset. Not upset, but I'm a little bit. I think. Look at this. This is horrible. But you see, I'm using OBS Studio. See, now that's basically without the green screen. Without the thing, it's just the green screen. I think it's also okay, I guess. No. Give me a second, please. Otherwise, I'm just going to leave it as it is. My face does look a little bit overexposed now. Why is it doing this? It's really strange. I will try the following. I'm going to try the following. I've got a second. I'm going to try to turn up. Okay. I turned up the brightness in my room a little bit. Oh my gosh. Sorry, folks. This is a little bit. This is a little bit disturbing. Please give me a second. I don't know why this is why it's doing that. Color of the shirt maybe, but actually there is no auto adjustment that I have here. I'm not doing any automatic color adjustment. Everything is actually I set the values. This is unbelievable. Otherwise, yeah. Last try. I think I'm just making the things worse here. You know what I'm gonna do? You know what? Who cares? Let's just leave it like this. I mean, if it's dark blue or whatever, I think it shouldn't make a huge difference. Yeah. So you know what? Yeah, it still breaks up a little bit every now and then. Okay. You know what? Yeah. I guess you don't mind. You don't mind the color. The thing that kind of disturbs me a little bit more is that my face is seems to be a little bit overexposed. But okay. Okay. Who cares? Who cares? Okay. Perfectionism is not the thing that we need now. So what I'm going to do now is therefore I would like to, before I throw out this water sample, I would like to check it. Okay. So that's the thing. I would like to simply check if there are any diatoms in there, and I need a pipette, pipette, and because the tip here is too narrow, what I do is I usually use scissors to cut off the tip so that I can also pick up larger samples. Okay. You can, of course, also use disposable pipettes. Yeah, but these are really, really nice and cool to use, so I kind of like it. You go into and you don't need a lot. The only problem with those sand samples is that because they're fairly thick, right? So this basically means that the cover glass is not really able to properly press everything flat. Yeah. So we're gonna spread it a little bit, yeah, and let's give it a try. Again, this is a little bit one of those last things that I usually do with specimen samples before I throw them out, flush them down the toilet, so to say. But occasionally there are a few interesting things in here. So, and you can see, yeah, it's reasonably flat. So, and let's check. Let's move over to the microscope view, scope view here. We got that. So let's put it in. And yeah, please lower your expectations, everyone. Okay. So if you're hoping to see a lot of living things in movement, you're probably gonna be disappointed. Lower your expectations, please. We see sand grains, obviously, and I'm going to zoom in and maybe we are able to see a few diatom shells. And I remember that at least still 1.5 years ago, there was still quite a bit of movement in here. Yes, so I go up with the magnification. Sand grains actually also look quite nice. Yeah. And looks pretty dead. I mean, look, one always has to be aware bacteria are still significantly smaller. I'm quite sure that there are plenty of bacteria in here that have survived. So saying that it's completely dead is a little bit probably not quite the correct thing to say. But at the same time, I also do not see a lot of diatom shells. Still a couple of months ago, I used to see quite a few of them. I wonder if they've also broken down, but actually those diatom shells are made of silica and they are quite stable actually otherwise. So I am indeed a little bit surprised that I don't see anything at all. Yeah. I wonder what this thing here is. Let me go up a little bit. Yep. Okay. Nothing of particular interest. What I generally also have once done is I've taken a video over, let's say, several minutes, and then I increased the speed by tenfold in a video editing program. And this then allowed you to see movements that were otherwise too slow to be seen. I mean, look at this here. Look, just look at this here. I'm going to show you. Can I move the arrow? There was just a short time ago, there was a little bit of movement here, and sometimes it's because there are some diatoms in many cases, or some other small microorganisms that are kind of pulling and pushing some of the sand grains, but there is actually not a lot of movement here. I am now, because somebody's asking, I'm using a polarization setup. Now it's called DIC. If you want to watch the whole thing in bright field, because most of you have bright field, so then it would look like this. Okay? So this one now also uses the natural colors. Sometimes this is also a good setting to use, because this way you're able to also see if there are green things around, because everything that's green is often or might contain chloroplast, or and do photosynthesis. Yeah, so, but I think we all agree that this sample did not survive 2 years. Oh, of course, I know that there might be different regions in the jar, maybe further up or further down, there might be different organisms. But generally, if there is life in there, then it should be easily visible. Yeah, so this, I think, summarizes everything here. I think I'm not going to further keep the sample. Yeah. So this was basically standing, for those of you just joined, 2 years on my on my desk, yeah, and no movement. Yeah. So the question, somebody's asking a question: what's the benefit of using polarized light? If you're using polarized light, then you can actually make well, it looks some specimens look very pretty when you use polarized light. Check out my crystal videos, you can use it when you use polarized light. You can also determine if there are certain crystals, for example, in a sample, or for example, starch grains or crystalline structures. So you can make certain things visible, which are otherwise not so easily visible. Yeah. So that's with a higher magnification, I doubt that a higher magnification would show them, because the atom shell. Look, there is movement. There is some movement here. Did you just see this? Yeah, there's something going on here. There could be some up. I bumped into it. I bumped into the mechanical stage. No. But usually, if there is, if you find one place where there's movement going on, then statistically, there should be others as well. Here we are again. Yeah? But I don't know what this could be. Yeah, I know that there are sometimes diatoms around that move, and they kind of. Oh, it's a diatom. I'm going now 60. Let me go up with the intensity. I think it must be a diatom, kind of hiding a little bit. I don't know. Yep, do you see this? Oh, here as well. Oh, here we go. Do you see this guy here? This long thing? It's difficult to see a little bit. because it's covered in all of this slimy, I don't know what it is, material. Yes, that's a diatom and it's alive. Here it is. Okay, I guess I have to correct myself here. Some of them were able to survive. Yeah. So that's a little bit of a thing. So what I'm gonna do is I'm gonna quickly go through again some of those questions, okay? And then as promised, I'm gonna move on. Yeah. Yeah, there are some questions here. Please apologize if I overlook some of them. There is a question about the micropipette capacity. Yeah, so I skip over many of them. So what's okay? Yes, I actually do have a recommendation. Okay, you're asking. I give you an answer. I want to quickly show you again the desk, because this is indeed I'm going to show you two of them. And just a second. I need to get some tips. And I don't have. Oh, I got one. No, I don't. Yeah, unfortunately, I don't have a tip for the large tip here. But I just want to show you quickly. They look quite the same size. Essentially, not sponsored, nothing. I just got them over Amazon. They sell the same pipettes using different brands. Okay, so yeah. Essentially, this one goes from 5 to 50 microliters. We focus here, and this one goes from 100 to 1,000 microliters, so basically 1 milliliter. And the tips are also, they're blue and they're bigger, but I don't have any more. I've got them, I guess, in the cellar, and I'm not using this one here. Okay, so I think the one that goes all the way up to 1,000 is too large, and I'm using the one from 5 to 50. And the reason I like this one over here is because the tips are much smaller, obviously, and also small enough that I'm actually able to pick up individual organisms from a slide under the microscope. Okay, so what I do is the following: I put a new tip on here, okay, and under the microscope, I actually go there and I'm able to pick up individual microorganisms. It's still a huge tip, okay? It's still compared to the size of the microorganism, but it actually does work. Yeah. So this is actually the reason why I probably would say 5 to 50 is fine. I was actually thinking about even buying a slightly smaller one. Yeah. And because sometimes it can be just a short trick. I'm going to show you a little trick. Sometimes it can be a little bit difficult to actually pick up individual objects here, because the angle is too flat, right? And then the tip is not submerged in water. Yeah. So what you want to do is you want to have an angle downwards. And look what I'm doing. This is gonna scare you. Yeah, I've got a lighter here. Okay, I've got to be careful that the heat doesn't damage the camera. I melt it. I bend it down. A little bit more maybe. I bend it down. You got to just make sure that just have to make sure that it doesn't that you don't burn a hole into it. And then when you put it on here, it's much easier. It's much easier for you to actually pick up samples this way. Okay. Yeah. I mean, if you want me to try this, I've actually burned a hole in here, right? But if you want me to actually show you how this actually looks like. Okay, then I'm going to simply try this again, so that you see, and let's try to maybe pick up individual items here. I don't know if it's so. Let's put it here. Okay, and let's go back again to the microscope view. This only works with a low power, obviously. Way too dense. Yeah, yeah. You see, this is kind of the droplet, and I don't know if this actually works now. So what you can do now, check what I'm doing. You kind of go in with a pipette like this, and you can try to actually, yeah, it actually did work. You can try to pick up individual things. I've picked up already tardigrades this way before, simply to separate them from all of the other mess, algae, sand grains, and so on that surround them. So, and I transfer it over into a, I'll just show you, let's go to a different empty place here. And then what I usually do is I go here again. Yeah, and then you get the idea, okay? Yeah. This I keep on doing that. I dilute it with water. I keep on doing that until, for example, I get only the animal, the tardigrade, for example, that I want to have. And I usually do that when I make videos, YouTube videos, where I just want to have the organism completely isolated and nothing that disturbs it around it. Yeah. So that's kind of the idea. Yeah. Of doing it like that. Yeah. So yeah, that's also something, yeah, that I essentially did not plan on showing you, but maybe it proves to be interesting to you. Don't know. Yeah, yeah. So let me quickly check again. The bottom right of this corner. Oops, I think I overlooked that. Has a full circular diatom shell. Oh, didn't see that. Yeah. So this, you know what? I might not throw it out right away. Maybe I'm going to indeed take some time the next couple of days to check out some of the things. And if I see some nice diatom shells, I'm going to pick them out. Yeah. So I'm scanning through the questions again. Micropipette capacity. Okay. Oh, gee, I overlooked it. Okay, you found some mounting medium here. Yeah. Okay. Yeah, some emailing issues. Yeah. Did you keep the sample wet or did you rehydrate it? No. I kept it definitely. I kept it wet. Yeah. As soon as you start drying it out, you end up you might end up killing a lot of living things here. Just a second, I have to, oops, this was my face that I deactivated. I wanted to activate now. Here we go again, okay? So sometimes the computer is a little bit overloaded with so many cameras. Yeah. When I connect a mirrorless Sony Alpha without an adapter, I get a reflection in the middle. That is, when you get a reflection, sometimes when you connect the camera and you get a bright spot in the center, sometimes this is due to the fact that the edge of the tube also reflects light. Köhler illumination would be the solution here. So this basically means that you have to really try to limit the light only to the place where that you're looking at. If there's too much reflection on the side of the tube, then this can actually cause a bright spot in the center, right? I don't know if there's an easy way to solve this. But yeah, that's the reason in most cases. Yeah. So I put a flexible silicone tube between the micropipette and the tip. I hold the tip in one hand and the micropipette in the other. Oh, that's an interesting one. So the idea is the following. Do I have a tube here? No, I don't have the tube here. But the idea was the following. I'm going to quickly explain this. That's actually quite a nice, thank you for the recommendation. What you do is you have a flexible tubing, for example, like in aquariums, you sometimes use those air tubes. And you connect it over here and the other one over here. And then what you have is you have a little bit more control maybe because you can keep it kind of separate. This is kind of the idea. Do you have to add water after some time or did you store it with the lid closed? I will tell you. wouldn't that potentially cause the buildup of anaerobic bacteria? So this is an interesting question, and the answer is not okay. I'll try to give you an answer. First of all, originally I kept the jar open. This causes, of course, water to evaporate, but that's a problem because if the water evaporates and this is salt water, then the salt water concentration goes up. So you do not want salt water concentration to go up above a certain critical level. So what I've done is then of course I added a little bit of tap water. But if you want to kind of prevent evaporation, then you add a lid. Now the question is if you add a lid, is it not going to become anaerobic? And the answer is no. The reason is because there is luckily still enough air in the jar. Yeah? And as long as you've got air, and if you occasionally aerate it, it's not a problem. The bottom part of the jar, or if the sand layer is thicker, then the bottom layers will become anaerobic. This also happens naturally on the beach, and that's why there are different microorganisms in the anaerobic part than in the top layer of the sand. I think it's too small here of a distance, right? So I think the whole thing would be aerobic still, also because there is a lot of air in here, right? So very briefly, if you now use a tube like this here to collect water samples, and I have done this as well, right? If you collect tubes like this, and if you want to, or traveling and you want to collect water samples, you really have to make sure that you do not fill up the whole tube all the way to the top with water, because then it is going to become anaerobic. Yeah, and that's a mistake that I've made some time ago. Is I collected water samples and I fill it up all the way to the top. No, not a good idea. And if you only keep it very small, a small amount of water with a lot of air in here, then honestly, it was able to store for days and no problem. But as soon as you fill it up and put a lid on it, it's gonna start smelling bad like sulfur because it starts to go anaerobic and it's gonna die off. Yeah? So that's simply I think an important thing what I would like to recommend. Yeah. So you can put a lid on top, do that, but make sure there is plenty of air. Okay, in here. Yeah. So that's so I want to move on a little bit. Yeah. So curl illumination, yes. Okay. So I've got a flexible silicone tube. We got that. So what I would like to do now is the following. I want to move on. It's already 43 minutes, and I would like to show you the next thing. So we talked about cameras. I put some old sand samples in here, and another thing that I did not try. I mean, this sample I did not try yet, but look what happened here. You know this one already from some time ago. Also last year or so I collected from a rainwater collected in the forest. There was a small temporary puddle with water fleas. I collected a jar and it looked brownish muddy, the water. This water here does not look brownish muddy anymore, it looks green. This basically means that algae started to grow in here. So this water sample I kept open. I always added more water when it evaporated. I just didn't feel like putting on a lid and I said, who cares? And I can always add a little bit of water, right? And this is what I have done. There are ostracods in here, floating around, swimming around in here. Not so many right now. Yeah. For whatever reason, a few. But the whole sample turned green, meaning that algae started to grow. Why all of a sudden? Because I left it standing on my window and there was a lot of direct sunlight. Normally you want to keep it out of direct sunlight. For example, if you've got aquariums and so on, you want to keep it out of direct sunlight because otherwise you promote the growth of algae. But that's exactly what I wanted. Yeah. So and here we are. And what I've done next is I have taken a tube, I filled it up, and I centrifuged it. And for sake of time, I'm not going to show you how I did that. But on the bottom here, you see this is a pellet. Yeah. And this is basically whatever concentrated stuff was in there and a little bit of water. And what I'm going to do is I want to put this under the microscope. And before I do that, I have to resuspend it. Okay, so what I do right now is I kind of tap it and I try to dissolve it, and this way I've increased the concentration quite significantly, and I hope that we're able to, hopefully, I don't know, find something interesting. Yeah? If this resuspending takes too much energy and time and patience, then we can also resuspend it using a pipette. That's also one of the reasons why they are very practical, because you can actually go all the way down, and simply by mixing it, by drawing it up and down, up and down. By the way, these micro pipettes, you can adjust the volume, in case you don't know that, by turning this here. Okay. So let's just go to the maximum. Yeah, so that's basically this is very concentrated stuff here, okay? So I've centrifuged this for about maybe 10 minutes or so. I resuspended it. You see it's a significantly dark, very dark green, so it's quite concentrated. And this allows me, if it's very concentrated, to use a fairly small amount. It's way more than I need. I can use a fairly small amount, and I can basically make a very thin layer and still get enough cells. So I'm going to make it deliberately very small. I'm going to put the rest back. Okay. I'm just going to put the lid on top so that I'm not going to spill anything. And again, a cover glass. There is when you keep water samples in your jar, there is in most cases always some kind of a progression, right? Certain organisms, microorganisms, are going to start to disappear, others will start to appear, right? That's a completely normal thing. So let's try this here. And you see that it's a very thin layer. That's good, because I really want to go up with the magnification. Why are cover glasses often dirty? Often, in almost all cases, unfortunately, I found out that they're dirty. And this is because of manufacturing, I guess. I don't know why. I have to tell you, I've got some slides here which are quite clean. And they're pretty high quality slides. But very often the cover glasses, they have, I'm just going to show you. These are directly fresh out of the box. Just to show you a little bit. Let me pick out a few of those. If you just kind of reflect them a little bit in light, you can sometimes see that there is a not so much here. But sometimes you actually see that there's a layer of, I think it might appear to be calcium carbonate, because when water was in contact with water, it starts to deposit, and then it makes those water spots. So I'm going to put this now, here. I yet have to find cover glasses that do not need to be cleaned. Well, only after, sometimes if I wash them first, then of course they're reasonably clean. But unfortunately, So this is how it looks like. Ooh, okay. Those fibers, maybe they were just contaminants. I don't know. It's way too. I don't see any. Look. Okay. Let's go up. I don't see anything yet. It's a little bit out of focus. There is stuff, of course, in here, but it is pretty small stuff. Look at this. Let me go up. We really have to go up a little bit with the magnification here, because it is pretty small. Wow, these. There, it is pretty small. Okay, let's use the natural. So I'm going to go up to 60 times. Look at this. I don't know. This is not Brownian motion. They are actually moving on their own. It seems to be a combination of Brownian motion. Look, there is some. Okay, this is 60. Those cells are pretty small. Okay. of course, they're now moving and streaming because of evaporation of capillary action beneath the cover glass. What was this? What's this? Do you see this? What? I have no idea. Could they be bacteria? Maybe some forms of cyanobacteria. Maybe some. I don't know. Yeah. There was this thing moving through the whole thing. Did you see? I don't know if you just saw that. I have no idea what this was. So I'm going to do the following. I'm going to turn on DIC to increase the contrast. So I'm now at 20 times. So the streaming motion that you see is because of the spreading of the liquid beneath the cover glass, right? So that's completely normal. But I don't know yeah, that could be indeed a ciliate swimming in another layer of focus. Yeah, but the layer, that would make sense. But the layer is so thin. Yeah, it's really compressed. Dark field is dark field is a problem. Dark field is the problem of dark field is that I only have it at low magnification, right? So this is why we're probably not able to see a lot. So this would be now dark field. Okay, so we're not able to see a lot because it's way too. Yeah, no, it's not good. Unfortunately. One of the things that I could try is this here is now phase contrast, but I think I lost it. No, here we go. That's now phase contrast. Also not the best. I think I'm just gonna stay with DIC or bright field, and then yeah. I have to close this here. I have to turn this up. So get all the adjustments, yeah. So, and maybe at 40 times. You see, I have to adjust it on yeah. Yeah, here, this must have been some kind of a fast-moving ciliate. Here it is. This guy's really fast. Maybe this one here right now in the center. Yes, that was it. What is this dark spot? Seems to be some kind of, I don't know, some dirt. Yeah, so in summary, the cells that we see here are pretty small. Considering the fact that they do look green, they're obviously greenish, they do photosynthesis. But more I cannot say. They do also have the size of bacteria as well. So they could be bacteria, photosynthetic bacteria, but I wouldn't be able to say. Just size alone is not indicative of anything. Yeah. Yeah. So this kind of explains a little bit, or not explains, but kind of clarifies a little bit of why the sample is so green, and evidently the ciliate also was able to survive the centrifugation process, yeah? Because the G forces are also kind of large, and not every organism survives it. But yeah, I have to also tell you that I do not consider this sample to be very interesting either, because normally if there are algae and there are larger algae. There are some really beautiful ones, and unfortunately we do not find them here. Some here as well. Yeah. So yeah, you get the idea. Could be more exciting, but on the other hand, we always learn a little bit here, right? I love the fact you didn't look alone before the stream, so you have to surprise with it. Yes, that's actually one of the things that I try to do a little bit. Is the following. It's a little bit one of the how do you say the the typical things here is that sometimes, and I'm guilty of this as well. I make those YouTube videos. I show you all these nice things, you know, and then the interesting and colorful. But what you do not see is that sometimes I've taken quite many videos before that, doing some video processing and so on. And I only show you the best things and the nicest things, but you do not see that there's actually quite a lot of preparation work behind it. For example, I mentioned it before, the tardigrades, right? If I show you a tardigrade moving along, it looks kind of nice. You don't see it like that in reality because the tardigrades are usually hidden behind some soil samples or some sand particles or some algae, and it's very messy, right? And if you want to get a nice video of them, you have to isolate them. So what I've spent quite a bit of time of sort of transferring those tardigrades to a new slide using my pipette, right? Until I get it in such a way that you actually see it very, very, very nicely, right? But microscopy, very often you have hopes and expectations and you don't see it nicely, right? Or there is nothing exciting there, or at least you say maybe it's not really exciting because it doesn't look cool, like this one over here. I mean, this is not so super exciting. But then on the other hand, it's still interesting to a certain extent, right? Because you see, wow, I didn't know that there are actually so many cells in there, and they're quite much smaller than what I expected. Occasionally I see some ciliates moving around, right? Yeah. So we still learn something. Yeah? So I'm trying to move a little bit the interest and the excitement away from this cool visual things to the actual learning, maybe. Yeah. Yeah. Okay. Semen under the microscope. No, I'm not going to do that. There are certain things that I don't want to do. Okay. What I would like to... I believe you're just taking care of the pond water samples and keeping them alive and healthy is a big job. It's not so difficult, really. But you have to add some water occasionally. And usually what happens is that there is always a progression happening. So that means certain organisms are going to disappear, others are going to appear, right? So that's a little bit a part of the normal thing, and then I occasionally put them again under the microscope. So what else did I want to do? Let's have a look at the time. Okay, it's 1 hour now. I wanted to show you, yes, because there have been questions about this as well, and I'm going to show you again. For some of you this will be new. I would like to show you some resources because right before the beginning of the stream, people wanted to know some identification resources, and unfortunately I cannot open the book for you because of copyright issues, and it says you're not supposed to do anything, spread So I'm going to just show you 2 editions of the same book, and there is, don't worry if it's in German, because this here is the old edition. And this is a really cool identification book for protists. Old edition, new edition. Thick and full of drawings. And I don't know, what does it say? 1,000 drawings or something? I have no idea. A lot of drawings. Over 1,000 drawings maybe. Very high quality drawings, and pretty much so far I found pretty much every organism that I found in the water sample I found in here. It doesn't matter if it's in German. Why does it not matter? Because if you have got the name, you identify the organism over the drawing, and if you've got the name, you can always Google for the name, and then you can get more information. Yeah. If you really want to read the text as well, you just get yourself for your mobile phone the Google Translate app. You take a picture of the text and it will translate it for you. Yeah? So the fact that it's in German should not deter you. Disadvantage, it's kind of expensive, the book, but maybe you can get the older edition, which is this one over here. These are serious books, okay? What I want to show you, however, let me quickly check where it is. I would like to show you, yes. If you go into Amazon, I'm in Amazon now, there is this preview of the book. And unfortunately, the preview of this book does not show you the main part with the drawings. However, there are a few drawings in there to give you an impression. Yeah, and you see that these drawings are very high quality. Yeah, and then this only shows, it's a table of contents, of course. Yeah, and then it only shows you the also beautiful drawings, and it only shows you some kind of the introductory text here. But this is not the main part. Yeah. So that's a little bit the thing. The later on, the main part is actually full of drawings. So there are, So on the left side you see the description, and on the right side you see the drawing. Yeah. So this is So this preview should not deter you. These are just some examples how the drawings look like. Okay, so this is basically from Amazon. It says here 44 euros. Let's put it this way: there are so far I really haven't found any other book that is that complete. Yeah, I also have to say that. Yeah, so this is just one thing. So if I would only get it if you're really serious about identification or about hobby microscopy and pond water analysis, and it does not only contain protists but other water organisms as well. Okay, so that is the first resource I wanted to show you. Okay, so this and there is we're waiting for your book. Oh yeah, I have in planning still. I have a long several things to say here, and another thing that I would like to show you, and this is where you get some free PDFs. However, also in English, and this is this web page over here from Wilhelm Feusner, feusner.at. Maybe I'm just gonna copy. I put the link, by the way, at the beginning of this live chat. Okay, at the beginning of the live chat, I copy pasted the link. I just want to show this to you here. Wilhelm Feusner, he was unfortunately passed away already. He was he was a very well-known protozoologist from the University of Salzburg in Austria. As a matter of fact, I personally, unfortunately, never got to meet him, but I've got some colleagues, biology colleagues, working with me in the same school. Who actually have known him personally, studied basically under him when they did their teacher training program in biology. He has published a lot, and some of his publications were he actually released freely online. Please be aware, those books, some of them are very scientific. Okay, some of them are really scientific. However, there's this one book that I would like to kind of show you, and it's called Freshwater Biology. And if you click on here, okay, it will open the PDF. So that's really, I really like this that he published, he made it all available. And there are identification keys in here that you can use. However, again, be aware that this is pretty scientific, okay? And the idea is like this, that you kind of start here at the top, right? And then you go down and either you branch off to the left or to the right, and based on the morphology of the organism, you can identify then what it is. Yeah. Have a look at this. However, the reason why I still think it's, even though it's good, is because there are still plenty of drawings in there that allow you to compare the things that you see with the drawing. And then once, if you say, it looks like a number 3 over here, then my organism looks like number 3. Then you've got the name down here, number 3. Yeah, this one over here, and you copy paste it into Google. You try to find a picture of it, a photograph of it, and then you can determine whether you've got a similar one, right? So this is the thing that I would recommend. Yeah, I mean, this is just overwhelming. Yeah. So here, for example, you can count the number of serial rows. Yeah, here right in the center. Yeah. And then if it's got 6 to 8, you go to and so on. You get the idea, right? Have a look at this. I tell you, for my purposes, it's not suitable because it is, I think, a little bit too advanced. Yeah, and not necessary because I'm just doing casual observation using the microscope. But I just wanted to show you that there are. I already think it's interesting just to show you what's out there. Yeah. Oh, come on. That's for example, a vorticella, right? For example, if it looks like this, then you know it's a peritrich. Yeah. So, and then the different kinds. Yeah. So maybe it's not even necessary to go through the whole identification tree, but you just basically go to the very bottom and you kind of look yeah, does mine look similar to which one does it look similar? Yeah. So it's yeah, just wanted to share this with you. Okay. Yeah, yeah. So I'm quickly going to go through the thing here again. Your channel is getting bigger. Ah, yeah, you might have to elect a few moderators for the chat stream. Yes, I was thinking about this as well. Yeah, I will soon receive Uprol and glycerol. Glycerol is easy to get, Uprol not. Yeah, I want to mount plant cells. Do you have any advices? Does it prevent stain like neutral red or methylene blue? I don't know really so much. Glycerol is slightly acidic, so it might change the dye a little bit. Uprol should be fairly stable. You have to try it out, I would say. Yeah. Generally, what I would say is do not overthink it too much. Try to get a stable mount together, and it doesn't really matter in most cases whether the dye or is stable over the next 50 or 100 years, right? I think that's not so. Yeah, for I would say for amateur purposes, not so important. Yeah. Let me quickly check. So what I would like to do next is the following. Yeah, so I just wanted to show this to you. I always have to keep track of a little bit of time. I'm now going to show you the most unpopular slide that I have. Why do I say unpopular? Because a couple of months ago when I received the slide, I made a video and it is my least viewed video. And I wonder why, and I think it's so nice. And I also actually showed you the slide before when I received it. But I just... This is a little bit the how do you say, the paradox of the situation, that the things that I consider really interesting, other people don't consider so interesting. Yeah? And it's this slide over here. And I just wanted to advertise again one of my older videos. This is an antique microscope slide, and it's one of the most expensive ones that I have. I think I paid over 30 euros for this one slide here. It's 100 years old around. Huh? And it is the tail of a monkey. And I really like this slide. I use it also for yeah, it's very educational because it shows so many different types of tissues. Yeah, and it's really well prepared. And yeah, unfortunately, maybe the video that I made was a little bit too theoretical. I don't know. I'm not going to go into a lot of theory. I just want to show it to you. And I'm going to put a link again to the video. Tail of a monkey under the microscope, I think I called it. Yeah, I just want to advertise this video because I don't want to believe that a slide which I consider so interesting was actually not considered interesting by, or at least the video. Or maybe some people don't like it don't like to think of the fact that actually it's the tail of a monkey that's under the microscope. I don't know. I'm just going to show it to you in any case now very briefly. Okay. Yeah. I'm going to go for bright field. I have to go in here. I have to focus. Yeah. And go down a little bit. Let's, yeah, I give you 4x, so just to give you a little bit of an overview. Yeah. And what you see is you see so many different types of tissues. Yeah. For example, just want to show this to you. Yeah. Again, this is one of the nice examples that if you don't know what it is, it says who cares, right? But actually, if you know what it is, it's very interesting. So, for example, what this here is, this is the bone, yeah, of the tail. You've got the bone marrow in between here, Here, that's 1, 2, 3, 4 muscle bundles. Basically, a pair of muscle moving the tail like this, the other pair moving it sideways. Yeah, because muscles are able to pull. Yeah? You've got over here on the side. Yeah, those things that you see here. Yeah, These are basically hair follicles for the fur. So you actually see that basically there was fur actually growing out here. Yeah. What you have surrounding here, these are fat, most likely fat tissues as well, which are kind of keeping the fur fatty and greasy to repel water. So there are lots of interesting things. What I thought, at least I thought interesting. This one over here, this guy over here. Look at it. Look at this. Do you see this here? This here. What is this here? It's an artery. It's a blood vessel. How do I know it's an artery, not a vein? Because it has such a thick wall. Yeah. You see over here again one of those fur hair growing out. Yeah? So I think this is a really nice slide. Again, 100 years old, properly prepared, and still very usable, obviously, and a very nice histological sample because it shows so many different tissue types. Yeah, and again over here you see that this again is a blood vessel here. Yeah, cross section. Yeah? So we can actually go a little bit in magnification if you want to. And yeah, see that the endothelium started to separate a little bit here due to the preparation. Yeah. Here, muscles again. Yeah. So just wanted to show this to you. Now here's cell differentiation. We debated last week. Yes, thank you. So many different cell types and tissue types. All these white things here look very much like fat, adipose tissue. All of the cells here have the same DNA, but they look so different. Why is this? Because of cell differentiation. Yeah? So I just wanted to again share this with you a little bit because it's just one of my favorite slides here. Again, 2 blood vessels here, but this kind of illustrates a little bit as well that if there is not a lot of movement going on, or if there's not a lot of color contrast going on, then sometimes it can be kind of abstract. And I once made the mistake. So never ever do that. I once made the mistake to actually show a nephew, one of my nephews. He wanted to see something under the microscope, and I showed him some beautiful histological slides. Never do that. If you want to get kids interested in microscopy, don't show them stuff like this, but actually show them some movement, some little critters, some worms, something exciting, right? That they can identify with. But this one over here, it's difficult to identify with. But I think it's so interesting as a biologist, So I just wanted to show this to you again, because it's sometimes difficult to share the enthusiasm, of course, if you if you have to explain what it is. You see, that's a little bit the problem with those slides. So okay, tail of a monkey, but what? Yeah, but if I'm saying, well, actually these are the cross sections of the fur, of the monkey trying to grow out. Right, and so then things start to make a little bit more sense. Here, for example, you see it quite nicely. How the fur is growing out. So just wanted to share this with you. And I would like to move on because I would like to still put the chocolate under the microscope. Okay, let me quickly check again the comments here. Can we see nerves in the slide? Let's put it this way. I wouldn't be surprised. I would not be surprised. But by the way, these are the tendons here. We ask, these here are tendons. Okay? But I wouldn't be surprised if there were nerves not in the center of the tail. I mean, there seems to be some marrow in here, but I wouldn't be surprised because in the spinal cord you also find them. So maybe there are nerves running through here as well, and there are plenty of nerves in here. But sometimes they are so thin, the axons, that they are difficult to see and difficult to identify. So that is the thing. Lots of cells in the muscle. Some of that probably. Connective tissue as well. Yes. Yeah. So for example, these things that you see here, those wavy things, most likely connective tissue. Yeah, connective tissue is made of collagen protein, and these are very strong fibers. Yeah, and they often run in parallel fibers, so this could quite well be connective tissue as well. Yeah? Is it possible to make a permanent slide with a cross section of a big insect? I mean, cutting a thin layer of a big insect, but I'm not sure if the slide would last long. Of course, this can be done. It has been done, and I have, just because you're asking, but I have one actually over here, several. There are insects, plenty of them. I have a problem seeing them without glasses. So that's a plant. I have, just saying, this is kind of, yeah. I just want to show you, cross sections do exist. Also hole mounts do exist. Got to be careful that that's another antique slide. Again, please do check my videos. I did make, how do you say, several videos where I show, just to illustrate this, yeah. They last long. If you do this properly, they can last a very long time. Yeah? This does not seem to be a cross section, but it seems to be more like a hole mount. But just for clarification, if you prepare them properly, they're properly dehydrated, properly mounted, they last indefinitely. Yeah. So I Nature Journal and brought my first microscope after watching my videos. I have no idea what I'm looking at, but I'm having much fun. Yes, honestly, it's very often this is the case that you see a lot of interesting stuff under the microscope, and you don't know exactly what it is. Yeah. In many cases, it could be dirt, dust, dirt. In some cases, it can be something interesting. Yeah. So, but I think it's still something, a hobby of discovery and observation. So what I'm going to do now is I'm going to now take those out because I'm pushing on, because I do want to show you the thing that I promised you. I have to always make sure that the slides are properly taken care of. Okay, because I really cherish them. So it's really actually surprising that those 100-year-old slides, the antique microscope slides, actually are of a higher quality than some modern ones, because the people at that time, they really spend a lot of time actually preparing them. They put some pride into it. And what I would like to do now as a last thing is the following. We're going to unpack this here. And I'm going to put this here under the microscope. Before I do that, I have to look. Do you remember this here? Yeah, there are still some of them are. These are the mosquito larvae from last week and 2 weeks ago. One of them is still around. Okay. Just saying. Yeah, they're still going. Maybe look at all the green spots on the bottom that you see here. Yeah, maybe also some kind of algae. So I don't know. So I need, in any case, I'm going to put this away. And I need the how do you call this, the stereo microscope, and we're going to unpack this here. And don't worry, a full video will follow. Okay, but because I'll be eating this stuff, not today, but yeah. So we're going to unpack this chocolate. This was a custom-made chocolate. Really, you can do that. I visited a chocolate company a couple of weeks ago. Yeah, and you see, it's all including a thank you card you could order. And look at this here. There's even a thing here. I typed it out, and they actually packaged the whole thing here as well. It's also a little bit of a thank you for all of your support. And this is how the chocolate looks like. And all of the ingredients that you see here, I could basically put together using an online app. And then they basically made the chocolate for me like this, and then you could buy it on the way out. So this is a pretty high quality chocolate, and what I'm going to do now is I chose those things here because I said maybe they kind of look nice under the microscope. I know I've got the plastic foil now over it, doesn't matter. So I'm going to put this under the stereo microscope. And unfortunately, the reflections here are a little bit of a problem. Yeah. Otherwise, I might actually, yeah. And there are some natural, how do you say, berries and I don't know, spices and whatever on here. You know what I'm going to do? I'm going to unpack it carefully anyway. Okay. Using my dirty scissors, maybe. I don't know. No. Let me show you the desk view again. I'm just going to make a little cut up. Not bumped it into here. I'm just going to make a little cut here. I'm going to carefully unpack it. So that we can have a quick look here at some of the chocolate. If I find it again here, it's much better under the microscope. So you can actually see that I could select some of the spices. I think it's peppermint or something. I don't know. I forgot. Yeah, there's a dried berry over here. Yeah, and we can actually zoom in a little bit. And we can now look at some of the things here under the microscope. And actually, I'm thinking about the following. Maybe I could try to actually get one of those leaves here and try to also put it under my compound microscope, simply by rehydrating it a little bit. Yeah? And then seeing how much of the cellular structure is still visible. Yeah? Yeah, but there's, for example, also these are some... Yeah, there's some sugar here as well, some dried fruits. Yeah. So quite a berry here. Yeah. Yeah, you get the idea. I'm just having a little bit of fun. But it's actually a high quality chocolate, obviously. Wasn't even that cheap. Doesn't matter, but in any case, this is a little bit of a how do you say, a motivation. You can put pretty much anything that you want under the microscope. Over here, for those of you who don't know this, I've got a stereo microscope. So basically, this is for observing objects that are opaque. Yeah, letters on the chocolate. You are surprised. You'll be surprised what they're actually able to mix into the chocolate. And one of the really nicest ones was actually chocolate. I'm not kidding. Where they put some chili peppers into it. It was actually chocolate that tasted spicy. It really tasted good. Okay, so essentially they have a huge variation of chocolate, and some of it, from very sweet to very, I don't know, almost bitter tasting, very raw chocolate to spicy even, but very good quality stuff. So that's also one thing that I wanted to show you. The combination of things that you actually have in the chocolate is kind of unusual. Yeah, so. So this is something I also just wanted to show you. I'm going to make a video of this, and I'm going to show you the whole thing here. Yeah? Let me put it here. Again, some of the questions. I hope I'm not going to overlook something here. I'm planning to cut a thin layer of the insect after freezing it to minus 20 degrees. In my mind, I was thinking it would be easier to cut. Yes, it's as a matter of fact, this is sometimes done. Objects that are difficult to cut can be frozen, but you have to be aware that if you want to cut it, then like microtoming it, then it might be a little bit difficult because the insect's exoskeleton is so hard. And for this reason, it might be a little bit difficult to cut. So maybe, but still, maybe even if you don't slice it, if you simply take apart the insect's different parts, then even they can be easier to observe. The legs separated from the antenna, from the wings, and so on. If you have just, even if you don't make a cross section through the whole insect, if you just separate those parts, it's going to make life a lot easier. Insect preparation as such is also a science in itself. To bleach the exoskeleton, you can use potassium hydroxide or lactic acid. There are different techniques here as well. So I'm going to skip ahead again. Okay. Chocolate with chili peppers is an ancient American drink. Ah, yeah. Okay. I think it was the ancient Maya civilizations who did that, but it was more like cacao instead. Yeah. So that I think I darkly remember many years ago I visited Mexico for summer holidays. I think I remember. Yes, I even ate chicken there with a chocolate sauce over it. And this chocolate sauce was, however, not sweet, but it was very good. I really loved it. Yeah. Have you made a microtome technical video? I would like to watch a tutorial about it. Yes, I have done a few of those. If you want, I can try some, show you some more. I'm using occasionally hand microtomes or table microtomes, but not those large sliding microtomes. Because if you want to actually make cross sections through like tissue, soft tissue, then the preparation time is quite extensive, and also it uses solvents that are not quite healthy. So I've not been doing that so much, but it is possible, and I can do that. I can make cross sections through plants. Just want to show you something else as well. This here is, I just switch over. Again, these are oak leaves that I put into alcohol for preservation. I would like to cut them in cross sections, but it's a little bit difficult to do. So I'm still working on a technique on actually doing that. Yeah, chicken mole. That's what it's called. Yeah, that's the thing that I've eaten. Yeah. So this is a little bit of thing is how do you actually cut those leaves? So what you do is you have to kind of support them by sandwiching them into some kind of a either a carrot or potatoes. They have the right consistency, and then you can try to use a hand microtome or a very sharp razor blade to cut across. Yeah, so this is a little bit a thing here, but embedding it in paraffin and then using a rotating microtome and so on is a little bit too much of a yeah. I don't have the equipment. Yeah, yeah. So that's something I just wanted to share with you. Okay. It's almost 1.5 hours again. So, folks, what I'm going to do now is I think I'm just going to, okay, that's just the microscope again. I'm just going to slowly call it quits. Then we're just going to save this for next week. What I've done here, and you can try this out as well, I've applied a little bit of glue on this onion here, and by peeling it off, you can actually put that under the microscope as well, and you should be able to see the surface structure. Last week I've already done this with an oak leaf. I've not tried it yet with a monocot plant like the onion, and I would like to see how the leaf structure here is different from those of the dicots, like for example of an oak tree. Yeah, you know what? I promise. Microbehunter, you said something about plant surface glue impressions on the description. You know what? Who cares? I'm just going to try to show it to you now. I promised, and we'll just add another 3 or 4 or 5 minutes. Okay. Yeah. So let's do the following. I need some tweezers. Okay, let's try this. Okay. Let's see if it separates even. So, you see the glue goes off. Okay. I'm just going to try to cut it off now. This goes here on here. Okay, this is just regular PVA glue, right? And again, I have not tried this yet before on the onion. So maybe it's gonna look good. Maybe it's gonna look I don't know, horrible or nothing, or I don't know. We're gonna see. Okay, that's a little bit. Yeah, I do see something, but let's see if this is something interesting. I do see something. Yes, and it looks interesting. Okay, those round things, these are bubbles. Forget about those. Let's go up with the magnification. Ah, yes. We see something. Yes, yes, we see something. We see something. What do I see here? So again, this basically that is glue. Okay. Yes. What do we actually see? Who could guess? What does this look like? Those things here. Okay. Any ideas what this could be? Stomata, yes. Yeah, these are the openings in on the bottom of the leaf that allow gas exchange to happen. Yeah. So this is something that we're also able to see here in a monocot plant like the onion. The reason why, you see it's not flat. That's why when I'm focusing, then some areas go into focus, others go out of focus. Yeah, the guard cells are basically those that are to the left and to the right of the openings in the leaves, the stomata. Yeah. Okay. So. Yeah, and here we see them again. So this is just something I wanted to share with you today again. I have used two different types of glue. Can show it to you as well. One of them is just regular white glue, wood glue. This works, and the other one is the clear PVA glue. Both of them should work. I've received basically success with both of them. Yeah. So, and I think I'm going to really call it quits now for today. What I'll be doing is I'll be adding, if you want to watch the video again or so, I'll be adding again some timestamps into the description, and hope that these things kind of also motivate you to experiment around a little bit with microscopy. Okay. It's a little bit of a new format where I'm trying to show you several things, one after the other. But if you join in next time, and if you join in a little bit earlier, then you can, of course, also start to post some questions in the chat in advance, and then maybe I'm able to answer them as well. For right now, I think I'm just going to... for right now, I'm just reading again. For right now, I just want to wish you all the best, okay? Do check out also the other videos of this channel if you're more interested in microscopes and cameras. Do check out maybe my other microscope channel called the Microbehunter Microscopy, which is the more technical one. And yes, another thing I just want to mention here. Ooh, almost forgot. I did make a couple of years ago podcasts, audio only, a little bit experimental. And if you go in this channel into the podcast tab, okay, I'll be publishing podcasts as well. And this basically means that there will be maybe a little bit longer and audio only. Okay, I'm just saying that just check them out. I'm still trying to develop different formats and experimenting, and you'll see. Okay, I wish you all the best. Okay, happy microbe hunting as always. See you around next time. Bye bye.